ApTOLL is a new kind of drug from the family of the aptamers. Aptamers are single strain nucleotides. So ApTOLL is one strand of DNA, very short. So this gives them advantages, very low immunogenicity. And also you can design them to bind your target, the targets you decide. So ApTOLL was designed to target the TLR4 receptor. So by targeting TLR4 and inhibiting TLR4, we are doing an anti-inflammatory effect. So we are stopping the inflammation early in the inflammation cascade, and this is supposed to give a neuroprotective effect. ApTOLL at the preclinical level shown substantial benefits in different species, in cerebral ischemia, in transient cerebral ischemia, but also in intracranial hemorrhages, and even in myocardial infarction. So there's a long story behind the development at the preclinical stage of ApTOLL. Given this background, we first started a first-in-human study with healthy subjects, and as there was no safety concern, we developed what is the APRIL trial, which is composed of two phases. Phase 1B is a dose escalation in which we tested 4 different tiers, including 8 patients in each tier. 6 received the active drug and 2 received the placebo. So after completing these 32 patients, the Data Safety Monitoring Board did not observe any safety issues, and they selected 2 of these doses to go forward to the next phase. And that was the highest dose and then the second dose. So the lower dose was really low as compared to the higher dose. And these 2 phases were pooled with placebos, or in the phase 2A, we recruited patients into either low dose, high dose, or placebo up to complete the whole study, 130 patients. The design was, as you can expect, what you could expect from patients that were candidates for thrombectomy with a documented large vessel occlusion in the anterior cerebral territory, either terminal ICA, M1 or M2 MCA occlusions that could be candidates or not to receive IVTPA. And in the interim between the imaging and the groin puncture for the thrombectomy, patients were consented and randomized to receive one of the 3 treatments. And from here, patients receive standard of care treatment with thrombectomy. And we performed an MRI at 72 hours to calculate the final infarct volume. And we assessed clinical outcomes, early clinical outcome with NIH at 72 hours and long-term at 72 hours. So the primary endpoints were safety, basically death, brain edema, hemorrhagic transformation, and stroke recurrence. And then we had 3 secondary efficacy outcomes, which were final infarct volume. Then we had NIH at 72 hours and long-term disability assessed with the MRS at 90 days. So in the primary analysis, we observed a reduction in mortality with the highest dose of ApTOLL. And this was statistically significant. We observed a reduction from 18% with the placebo to 4% with the high dose of ApTOLL. We also observed trends to better outcomes, also non-significant, in terms of lower rates of symptomatic intracranial hemorrhages and less brain edema. So that was very encouraging. And on top of that, in the efficacy analysis, we did have significant results in terms of lower final infarct volume on the MRI at 72 hours, less neurological impairment at 72 hours, so lower NIH, and a clear shift towards less disability at 90 days with a common odds ratio of 2.61. And that was with a high dose, basically. So all the results were aligned and confirming that there might be a positive biological effect of the drug with effective or positive measures in all these outcomes. There was a lot of excitement, mainly because it's for the first time a neuroprotection trial shows a positive is positive, and in the primary endpoint and the secondary endpoints. Also, as I said, it is consistent in all the endpoints, so that is very reassuring. And the obvious questions that we got more times is, what is the next step? And the next step is, we have two steps. First, we planned already to start a trial in which we will start administration of ApTOLL at the pre-hospital level in patients with suspected severe stroke, in which EMS or paramedics suspect a stroke. They will be randomized and will start treatment in the field in a randomized trial. The objective is to learn if it is feasible and how much it might be positive to advance initiation of neuroprotection. This trial will include obviously patients not only with large vessel occlusions, also patients with ischemic stroke without a large vessel occlusion, but most important, patients with intracranial hemorrhages. And we'll have a first glimpse of maybe an effect in patients who are experiencing intracranial hemorrhage. And second step will be towards a larger trial, a confirmatory pivotal trial that will lead to potentially to the indication by the agencies of the treatment. And this we are targeting the last quarter of the present year to start the recruitment in the study, in which we may assess also the efficacy of a higher dose, an even higher dose that might have even a higher effect.